Have you ever wondered what really happens when your DNA mutates? Or would you like to know which factors mutate your DNA? Or, for my fellow biochemists, would you like to see the mechanism by which DNA mutations occur? That's what we will talk about today, but before we go any further, hello, my name is Bara. I'm a second-year biochemist at Oxford University, and I guess this is my first scientific video. So can your DNA at this point in your life mutate? Yes. And when does it happen? Every single second of your life. Actually, as I was saying the last sentence, your DNA probably mutated about 30 times because according to Harvey Lodish, a famous textbook author, our DNA mutates about 10 times per second. But other sources quote numbers that are similarly high and usually based on the type of mutation. So I remember our lecturer quoted last year something like 55,000 single-strand breaks per day and 12,000 depurination events per day, and we will talk about these type of mutations, but the key point is that our DNA mutates all the time and with no apparent reason. But why? Well, it turns out that air, sunlight, even the food that we eat contribute to DNA damage, which is pretty freaky to think about. Honestly, probably even more freaky than all the hoaxes that we heard last year about viruses, vaccines, chips by Bill Gates. But let's turn away from that. I promise this is evidence-based, so let's talk about DNA damage. DNA can be damaged by a number of ways, and several different types of mutations can be a result of that. Today we will talk about 3 types that I just picked because, I don't know, I like these types of mutations, I guess. And those are point mutations, single-stranded breaks, and interstrand crosslinks. So in our DNA, we have only 4 building blocks, A, T, C, and G. And what we need to know is that A pairs with T and C pairs with G. And I hinted at the fact that DNA can be damaged by seemingly innocuous things, as innocuous as water. Then there are things that are less innocuous. Cigarette smoke, for example, contains 9 different types of carcinogens. For instance, here you can see a mechanism by which a compound named NNN, N-nitrosonornicotine, mutates our DNA. This leads to a so-called DNA adduct, which changes the base pairing between these building blocks. Here, G cannot bind C anymore, and I haven't indicated it in this drawing, but in this case, G would rather base pair with T, not C, and that's really bad. That's a mutation. Something that's as bad, and in some cases way worse than a mutation, is a break in a strand of DNA, and those kind of breaks are usually caused by radicals, and radicals are generated by sunlight, by our metabolism. It also happens all the time, unfortunately. Lastly, I want to show you what can happen when we ingest alcohol, because alcohol is just a precursor to aldehydes, reactive aldehydes that can react with many different types of macromolecules and create crosslinks, even in DNA. And crosslinks can be very bad. They can hinder DNA replication, DNA transcription, DNA repair, all those things that you don't have to worry about, but it's a very bad DNA damage. Okay, so at this point you may be asking, if this happens every single second in my body, how am I still alive? And how serious can mutagens in the sunlight, in alcohol really be? Surely smoking is not harmful if my DNA mutates anyway. And this is indeed a good point, something that my grandma would say, but it is not correct. And we will talk about the reasons why we can get away with quite a lot of DNA damage, but we will also talk about the reasons why some people develop cancer more rapidly, accumulate mutations more rapidly, age more rapidly, and why others don't. So one reason why we can get away with this much DNA damage is something called junk DNA. And the other reason is something called DNA repair. So junk DNA. Junk DNA is just the DNA that doesn't encode any protein. And that might seem counterintuitive to most people who are taught at high school and even elementary school that DNA is just the information for encoding proteins. But actually, this kind of DNA is only constituting about 2% of our genomes, and 98% is this so-called junk. It would then be quite rational to assume that our proteins won't be affected by any kind of mutation that damages our DNA, or at least there might be a pretty big chance. But unfortunately, that's not the whole story, because of the packaging of DNA and because of the fact that those 2% of protein coding DNA is usually more accessible, more open, and those 98% are more closed, more inaccessible to any kind of external damaging or not damaging agent. And so what happens when those 2% of our DNA mutate? Well, then we can start talking about cancer-driving mutations and aging. So, cancer. As we saw, when our DNA is damaged and not repaired, a mutation occurs. And cancer is really just a collection of diseases that are caused by a mutation in specific genes. These genes control growth, division, migration, and other things, but these are the most important, so that when such a mutation occurs, the cell loses its control. It starts to divide like crazy, it forms tumors, it scrambles its chromosomes, which is pretty cool. Not really cool, but it happens. Mutations are not the only proposed cause of cancer. Epigenetic marks or chronic inflammation have also been linked to cancer in this way, but their mechanism of action on the molecular level is way less understood. Anyway, let's get back to the topic. Now, how many mutations are required to turn a normal cell into a cancer cell? Well, in 2017, Dr. Peter Campbell from the Wellcome Trust Sanger Institute addressed this quite long-standing question, and it turns out that it only takes between 1 and 10 mutations to turn a normal, perfectly functioning cell into a cancerous cell. And this research group also established that these cells, before turning cancerous, already accumulated tens and hundreds of mutations. So we can't tolerate DNA damage, and that is because we have quite a lot of junk, although that's not the main reason, and because we have DNA repairing pathways, which is the topic of the next section. However, it only takes one mutation for a cell to become cancerous, and keep in mind that we have trillions of cells in our bodies, 37 trillion to be precise, according to some studies, and all of these cells acquire DNA damage. Let's now turn to the other defense against mutations, which is DNA repair. And, of all the beauty in the molecular world, I think DNA repair pathways remain the most fascinating topic to me. And the reason why I find it amazing is that we are approaching a point in time when we can just look at molecules inside of our bodies. And in the case of DNA repair, there are tens of proteins that interact and move around and all have their roles. So to give you an idea, this is how crowded the Fanconi anemia repair pathway is. And let's ignore the repair pathway itself because it's unnecessarily complicated for a silly little video like this. But just look at these proteins. Look how cool they are, and they look like this. We know that for sure right now. And just for a brief context, Fanconi anemia is the name of this repair pathway because a mutation in any single of the 23 genes involved in the pathway causes Fanconi anemia. And children with this inherited genetic disorder suffer from cancer, from organ failure, and often die very young. I think it proves how serious mutations really are and how lucky we are to have DNA repair systems that evolved to a near perfection. But a mutation in a DNA repair gene acquired during lifetime, so not inherited, has been linked to cancer as well. When DNA cannot be repaired, the cell decides to commit suicide. And this suicidal process is called apoptosis. And when apoptosis is defective as well, the cell has no other choice than to leave DNA unrepaired, which then leads to cancer as well. So what I really wanted you to know is that DNA repair is very, very important. So why do some people get cancer and others don't? Well, it's kind of obvious when we think about it. We defined cancer as a collection of diseases caused by driving mutations. And so the more carcinogen we expose ourselves to, the higher the rate of mutations, and the more mutations and the more cancer-driving mutations we accumulate. The most obvious example of that is the correlation between the incidence of lung cancer and the fact that someone is a smoker. And an article from 2016 in the Murray and Nattles textbook of respiratory medicine states DNA ADEC levels correlate with daily or lifetime cigarette consumption and do not reverse after smoking cessation. It's not that simple. It's never simple with cancer, but the changes in coding DNA and its expression have with no doubt been established as the leading cause of cancer and as the leading cause of age-related diseases in general. And when you think about it, cancer is kind of natural. It's not any sort of pathogen that's trying to attack us from outside. It's born out of healthy cells. And so everyone is destined to have cancer because everyone has DNA that mutates, and that might change very soon. People like David Sinclair and other researchers in the aging field are making very rapid advances, but at this very stage of medicine and science, everyone will get cancer sooner or later. So the question really becomes how to avoid cancer for at least another 200-300 years, which ultimately is the same question as how to increase our health span, a part of our lives in which we are generally in good health. We've already seen the obvious things like quitting smoking, getting away from the sunlight, using sunscreen. Of course, these things are full of mutagens, so let's protect ourselves. But then there's the category of the less obvious and even counterintuitive things that include heat exposure, cold exposure, and mostly diet and exercise, which are a bit of a barbed wire topic, but we will only talk about caloric restriction today, but hopefully maybe I will do another video. Although we all know that we need to eat in a healthy way, we need to exercise in a healthy way, what does it really mean? We won't know until we have sufficient scientific evidence and until we really understand the molecular mechanisms behind longevity, behind cancer resistance. Fortunately, there's one lifestyle change that I think is safe to discuss, and that is caloric restriction without malnutrition. A study in Nature from 2016 states, dietary restriction is the best documented intervention for extending lifespan in numerous species and retards many symptoms of aging. Quoted are 3 studies, and the first one I think is the most mind-blowing. The title is Caloric Restriction Delays Disease Onset and Mortality in Rhesus Monkeys. This is a 20-year study investigating the lifetime effects of caloric restriction, which was introduced in adulthood. Not, babies shouldn't be calorically restricted. This was introduced to monkeys that were 5 to 7 year olds, and ever since then, these monkeys were either given the amount of food that they usually ate up until that point. That was a control group. And in the experimental group, each animal's food intake was reduced by 30%. And that's it. That was all that the people did to these monkeys. 20 years later, this is how a control animal looked like. We can see that their face is pretty pale, grayish hair, hunchback, a better posture overall. This animal is 27 years old, And because an average lifespan of rhesus monkeys in captivity is 27-30 years old, we cannot really blame the researchers for the fact that this monkey looks really old. It's supposed to be kind of old. It would correspond to people who are 70-80 years old probably. However, a 27-year-old animal on a caloric restriction diet looked like this, and that's pretty good, right? Clearly, this animal looks much younger. It has much more hair that has retained its color. The posture is much better. Even from the animal's gaze, I think we can see that it's still alert and active. And I think it's pretty safe to say that it looks like there's much greater age difference when in reality there's no age difference. And although the animals on CR look subjectively much younger than the control group animals, that's not really what this research paper was all about. This research paper looked at the biological markers of aging, and they really wanted to determine whether these CR animals just looked younger or whether they were actually biologically younger than the control group animals. Which means that they looked at the age-related conditions that are most prevalent in monkeys and in humans, because there's really no difference between humans and monkeys. So they looked at things like cancer incidence of cardiovascular diseases, and what they found out was just mind-blowing. In the control group, 8 animals developed cancer at various stages of life, whilst only 4 CR animals got cancer. And in both cases, the most common cancer identified was gastrointestinal adenocarcinoma, so stomach cancer. Similarly, 4 control animals were diagnosed with cardiovascular disease, and only 2 CR animals had the same diagnosis. Therefore, there was 50% incidence reduction of these 2 diseases, which are the most prevalent age-related diseases in monkeys and in humans. The final and the most amazing result of this study was on diabetes, because 16 animals of the control group were classified as diabetic, 11 pre-diabetics and 5 diabetics. In contrast, no CR animal developed diabetes and maintained a great metabolic function throughout their whole lives. Needless to say that 50% of the control animals died at the end of the study, as opposed to 30% of the calorically restricted animals, and 37% of the control animals died of age-related diseases, whilst only 13% of the animals that died with caloric restriction died with age-related diseases. And keep in mind that all of these monkeys were fed a nutritionally adequate diet. It was a low-fat diet in particular, but for our purposes we can just say it was a healthy diet. And that means for us humans, keeping the amount of refined and ultra-processed foods to minimum, or really just close to zero. And just a quick note, because this is a topic that's very dear to my heart, I'm not a fan of any animal studies, especially vertebrate studies, and I do firmly believe that the science really needs to start moving away from that, and hopefully in my lifetime things will change. So how does this work? Why do reduced calories increase lifespan and delay age-related diseases? Well, caloric restriction ultimately leads to low fatty acid levels and low glucose levels, and that's no matter what the type of the diet is, because our criterion is sufficient nutrition. A hard carb diet wouldn't do, because we all need that those essential amino acids, those essential fatty acids. Levels of glucose and fatty acids dictate which hormones are going to be released into our bodies, and in this case these would be glucagon and adrenaline, whilst insulin would be kept low. Hormones then dictate the rest of the biology, and the most important consequence of these hormones is the activation of sirtuin proteins. Sirtuins have been found to be expressed generally in response to stressors like high heat, low calories, and what doesn't kill us makes us stronger, or At least that's certainly the case with sirtuins. Stress-induced sirtuins increase DNA repair by recruiting DNA repair proteins and by controlling chromatin accessibility. They also decrease inflammation by deactivating pro-inflammatory transcription factors, and they decrease tumorigenesis by acting to suppress cancer formation. However, in one study, sirtuins have been found to be hijacked by cancer itself, driving the growth of already established tumors. So while there is sufficient evidence of sirtuins preventing cancer, there is also a suggestion that if a tumor is already present, sirtuins can act in its favor. The evidence we have for this, for sirtuins mediating longevity effects of caloric restriction, comes mainly from mice, because with mice we can do more than longitudinal studies. We can delete specific genes, and that is especially revealing. When we delete a gene, we can see which function goes missing together with that gene. Such mice with deleted genes are called knockouts. And so when we look at sirtuin knockout mice, we see premature aging, tumor formation, no benefit from the caloric restriction diet. A final thing to say, just to make this video complete, is that sirtuins are not the only molecules of longevity. For example, mTOR is also strongly associated with aging, but in the reverse direction. So low levels of mTOR have been demonstrated to increase lifespans of organisms as diverse as yeast, worms, flies, even mice. So high levels of branched amino acids lead to high levels of mTOR and shorter lifespan. So to wrap up, we've talked about how mutagens damage our DNA, how that's kind of okay because our bodies can usually repair this. We also talked about the link between mutations and cancer, the chief age-related disease, which can be delayed by the action of sirtuins that are triggered by caloric restriction. Wow, I really thought that I'd never finish this, but yeah, thank you so much for watching. Please tell me what you think, whether this even made sense, and the quality will improve, I promise.